Good afternoon. So, first of all, I'd like to thank the organizers for giving me the opportunity to present our work. So, our work is titled CDGCN, Conditional de novo drug generative model using Graph Convolution Networks. So, first I'll be giving the introduction and the motivation behind this work. So, our work involves AI in drug discovery. So, drug discovery is the process of finding effective molecules which reduce the effect of a disease by binding to a target protein. The pipeline of drug discovery or drug development looks like something like this. So, the first stage is the drug discovery phase, where basically we search for effective compounds, and the search boils down to approximately 10,000 compounds. After this, some preclinical research and clinical trials are done, and the number of compounds boils down to a few. Finally, single compound is approved for use as a drug. The first stage in this pipeline, that is the drug discovery phase, is quite expensive and time-consuming because of the enormous search space of compounds. And that size of the search space lies between 10 to the power 60 to 10 to the power 100. Using artificial intelligence can help in making the search more efficient and informed, and using artificial intelligence, we can also go beyond searching for known drugs and enter into the territory of de novo drug generation, which basically means we now search for novel drugs which are not yet existing. So, the state-of-the-art methods for de novo drug generation, we observed two state-of-the-art models, and both of them utilize deep learning approach. So, the first state-of-the-art method was proposed by Lee et al. In the year 2018, and they proposed a graph generative model for protein-specific generation of novel drugs. So, this model uses a one-hot encoding of a protein as input and gives as output a drug structure. This model requires existing drugs of known proteins for training. So it is trained on like protein drug pairs, and the larger the number of drugs for any protein, the better this model generates drugs for that protein. The limitation of this model is that it does not generalize to generating drugs for novel proteins for which there is no information regarding existing drugs. The second state-of-the-art method deals with de novo drug generation for novel proteins, and this method was proposed by Grechishnikova in the year 2021, and this is a sequence-to-sequence generative model which basically translates a protein sequence into a drug SMILES sequence. However, the limitation of this model is that it overfits quite easily on the training dataset, and it produces significantly fewer number of unique novel molecules for novel proteins. So, our contribution, CDGCN, addresses the limitations of the previous state-of-the-art methods, and we address the task of generating novel drugs from scratch for any given input. So basically, our model, CDGCN, takes as input a protein sequence and gives as output a drug structure. And that structure needs to have drug-like properties like chemical validity, uniqueness, novelty, and a good binding energy. To go a bit deeper into the approach, as I mentioned, the input to our model is a target protein sequence, and that protein sequence is passed through a pre-trained protein representation model. This model is trained on the task of protein function prediction. So whatever protein encoding we obtain from this model has meaningful features which are useful in predicting the function of a protein. After we obtain the protein encoding, using that protein encoding, we define the graph generative process, the drug graph generative process, which is an iterative process, and it essentially grows the graph in a sequential manner. So the first step of the graph generative process is the initialization, where we basically add the first node to an empty graph. And the first node, the probability of what node that will be, it depends on the protein encoding obtained from the previous one. In the sequential generative process, at any given step, we basically pass the intermediate graph structure into a conditional graph generative model, which then takes the intermediate graph structure, encodes it into a graph encoding, and from the encoding, it then decides which action to take in the current step for the next step. The next action can be any of the three. So one action can be append, which means that it adds a new node to the intermediate graph. It could be a connect action, which means that it adds a new edge between two existing nodes in the intermediate graph. Or it can be the end action, which means that we terminate the graph generation process, and whatever current intermediate structure we have is then added to the list of the generated structures for the input protein. So the conditional graph generative model consists of two parts. One is the encoder, which is responsible for outputting a graph encoding for the intermediate graph structure. And the second part is a decoder, which is responsible for deciding the next action in the generative path, taking the graph encoding as input. So the encoder, which encodes the graph encoding, is based on the graph neural network architecture, which basically generates the vector representation for every node in the graph, depending on its neighborhood information. So for obtaining the encoding of every node in the graph, we basically perform an iterative neighborhood aggregation where the information from the neighbors of every node is collected together, and then the node is updated with that information. After obtaining the node encoding for all the nodes, we then combine all the node encodings into a single graph level encoding, and that is known as a readout. The parameters of the GNN are optimized based on the loss function defined for the final task, that is the generation. So going a little bit more deeper into the methodology, so initially we pass protein sequence as input to a pre-trained representation model. We obtain a vector representation for the protein sequence. Then, using the vector representation, we define the probability scores for the initial node to be added to the empty graph. At any given point in the sequential generative process, we pass the nodes, the nodes of the current intermediate graph are encoded using L graph convolution layers and subsequent feed-forward layers. So basically, the idea is that at every generation step, we take the node encoding and concatenate the protein encoding with it, which allows us to capture the interaction between the protein and the intermediate drug structure at every generation step. Finally, we aggregate all the information and pass it through the decoder to decide the next action. After the end action is selected, we obtain the final structure and add it to the list of generated molecules. CDGCN is trained using by minimizing the negative log likelihood of generating a molecular graph using a particular generative path given the target protein. For evaluating our model, we we used two data sets. One is the ChemBL data set, which we use to pre-train our models and our model, and it consists of 1.5 million molecules. The second data set is binding DB, which consists of protein ligand pairs, and we use this data set to fine-tune our model for the protein-specific drug generation task. For evaluating, we utilize these metrics, which is a validity, which means that what percentage of the generated molecules are actually chemically valid. Then novelty, which means what percentage of the generated molecules are novel, and what is the time required to generate a particular number of molecules. Apart from this, we also evaluated the QED and SAS scores for all the generated molecules and the binding energy of the generated molecules for a particular protein. Here, the table describes the binding energy of generated molecules for 5 test proteins, and these proteins were not seen by the model during the training. So we observed that our model, CDGCN, could obtain a better binding energy for the best generated molecule compared to the known ligand for that protein. For example, if I take the first protein, which is 4NOS, then we see that the best generated molecule could achieve a binding energy of -8.6, whereas the known ligand has a binding energy of -7.9. We also observed that compared to the state-of-the-art Graph-Convolution, CDGCN gives a better binding energy for the five test proteins. CDGCN is also very efficient in time. So it can generate almost 1,000 molecules, novel molecules, in the time frame of approximately 2 minutes. So it is very comparable to its graph model baseline, Lee et al. However, if we compare it with Graph-Convolution, the time difference is much drastic. And CDGCN generates novel molecules for novel proteins very quickly compared to the state-of-the-art. The generated molecules are also very easy to synthesize, and their synthetic accessibility score is better as compared to the state-of-the-art. We could also observe that CDGCN is capable of generating more number of novel drug-like molecules, which have a good QED score compared to the state-of-the-art. And I would like to point out that almost more than 99% of the generated molecules for our CDGCN approach is 99%. Here I show two examples. So on the left-hand side, the image consists of the result of docking for a generated molecule by CDGCN and the protein integrin alpha L, and the docking result, the binding energy between these two turned out to be -7.3. On the right-hand side, I show another docking results between the protein vascular endothelial growth factor receptor 2, that's a big one, and the generated molecule by CDGCN tagged as G, and the known ligand for the protein tagged as L. So the known ligand gave a binding energy of -11.1, whereas the generated molecule gave a binding energy of -11.2. To conclude, the molecules that were generated using our proposed method CDGCN were able to give better binding affinity and drug likeness compared to its state-of-the-art, and in some cases, the molecules could also give better binding affinity to some of the known ligands for the proteins. Our proposed method is much faster in computation time as well compared to the state-of-the-art for novel drug generation for novel proteins. As part of the future work, we aim to design de novo drug generative model which can target a group of proteins. So it means that we are able to generate novel drugs which target a group of proteins rather than a single protein. Secondly, we also want to explore more graph neural network methodologies which address the problem of graph isomorphism, which the current model does not. And addressing this problem could better distinguish between molecular structures. Finally, I would again like to thank you, the reviewers, for giving constructive feedback on our paper and helped tremendously to improve upon it. So, thank you.